The broadcast is now starting. All attendees are in listen-only mode.
Good midday to everyone. This is Sherry Lynn Phelps. I'm the Agronomy Manager with Saskatchewan Pulse Growers, and I'll be the moderator for this webinar. We also have Andrea Lauder, who is our Communications Manager, and she's done all the heavy lifting in terms of organizing the webinars and looking after functioning administration. Without her, the webinars just wouldn't be possible. A big thank you to Andrea. Now to our participants. A big welcome to the first Pulse webinar for 2019. We have 2 more scheduled that can be found on our website under News and Events tab. A bit on the administration before we get started. To get CCA credits for today's webinar, you must be watching it live. For those who attended the webinar, Andrea will send out an email after the webinar is over requesting your CCA number. At this time, you can respond with that number. If more than one person is watching from one computer, you will need someone to verify those in attendance, and all can receive CCA credits. Webinar will be recorded and posted to the Saskatchewan Pulse Growers website for future viewing for those unable to attend or for those who want another look at what was addressed. Videos of the webinar will be posted to our website under the Communications tab. For today's webinar, all participants will be muted. We will be happy to take questions. To ask a question, please type it into the question box located in the GoToWebinar dashboard. For today's webinar, Dr. Chatterton has divided her talk into 3 sections, and we can address questions after each section as well as at the end of the presentation. Today we have Dr. Syama Chatterton speaking to us from Agriculture and Agri-Food Canada in Lethbridge. Dr. Chatterton joined the Lethbridge Research Station in July of 2011 as a pulse and special crops pathologist. Her research focuses on management of root and foliar diseases of pulse crops and molecular diagnostics and identification of soil-borne pathogens. She currently leads a number of research projects on pea root rot, white mold of dry bean, and chocolate spot of faba bean. Besides her passion for pulse diseases, she also has a passion for cooking with pulses, and particularly trying all sorts of different tricks to get her kids to eat them. She will be providing an update on her current research on Aphanomyces root rot, starting with a summary of the results of 4 years of the pulse root rot surveys in Alberta and Saskatchewan. She will then provide an overview of her lab research to determine threshold concentrations of Aphanomyces and field distribution. Finally, she will discuss challenges and lessons learned from the field trials aimed at evaluating management options for Aphanomyces. I will now turn the controls over to Dr. Chatterton.
Okay, great. Thank you, Sherilyn. So I'm going to start off here with an overview of my presentation. Okay, there it goes. It was stuck for a second. My computer's woken up. Okay, so I'm going to start with an overview of my presentation. And as Sherilyn said, I'm going to break this presentation up into 3 parts with time for questions after each part. So I'm going to start with talking about the summary of the survey results, pause for questions, and then I'll talk about my research projects that are ongoing in the lab. I'll pause again for questions after that. And then we'll talk about management options, and particularly as these pertain to field trials and what we've learned about some management options over the past few years. So I'm going to start talking about surveys. And I've presented on surveys for a number of years, and today I'm going to provide a wrap-up summary of those surveys and the 4 years of results and what we've learned from them. The purpose of the surveys were to determine the prevalence and distribution of Aphanomyces root rot, and at the same time determine what other pathogens are involved in the root rot complex and what their distribution was across the prairies. So I'll start with an overview here with a map showing really the results in a concise way, the results of our 4 years of surveying. And anywhere you see a red pin is where we found pea fields that were positive for Aphanomyces, and then the yellow pins are lentil fields that were positive for Aphanomyces. So this quick snapshot here shows that Aphanomyces is really present wherever peas and lentils are grown. We see it in the Peace River region of Alberta, all the way down to the southern regions, close to the border with the US, and we see it from the western edge of production all the way out into Manitoba. And now I'll break it up into what we saw over different years. And so these are the Aphanomyces positive pea fields that were detected in Saskatchewan from 2015 to 2018. What I've done here is shown prevalence, so that's the percent of fields that were positive for Aphanomyces. We see those in the purple bars, and then the blue bars here is the percent incidence, or the percent of samples that were positive for Aphanomyces. And I wanted to link this with weather patterns that we experienced over these past 4 years, and you can see that here by this light green, these light green dots. And so the first thing to notice is that 2015, 2017, and 2018 were fairly dry, and a mean precipitation across the province, which is a really big area to try and find the average, but mean precipitation kind of hovered about 150 millimeters in the growing season. So that's from May to August. And then 2016 was quite a bit of a wetter year. And what we can see is that for the most part, that prevalence and incidence numbers are following that precipitation level. The prevalence numbers here in 2015 are a little bit skewed, and that's because it was the first year that we did the surveys in Saskatchewan, so we were particularly targeting fields that we thought might have a problem with Aphanomyces. So 2015 wasn't a random survey, it was a targeted survey, and that's why we see this high prevalence in 2015. But we can see that the incidence was actually fairly low. In 2016, was a wet year, and it was nice to have one wet year in the survey period because we can really see what that impact is of moisture and how that affects the expression of Aphanomyces root rot. So I'm going to move on to lentils, and for Saskatchewan lentils, we only surveyed in 2016 and 2017, so we don't have that full 4-year view, but I'm going to show you the results from Alberta as well because we did 1 extra year of surveying. But we can see very similar patterns. 2016, that wet year, we saw fairly high prevalence of incidence, close to between 60 to 70%, and then that drops off in 2017, which was that dry year with our max prevalence here of about 40%. And like I said, I'll show you the Alberta results here. This was quite dramatic in 2017, where we actually didn't detect any positive lentil fields at all in 2017 because of that dry season. Alberta tends to be a little bit, the Alberta lentil production area does tend to be a little bit drier than Saskatchewan lentil production area, and that's why we really saw that there wasn't a big impact of Aphanomyces root rot in lentils in 2017. But if people have heard me talk about root rots before, I think it's important to note that Aphanomyces is not our only problem. And so what I've done here is summarized all of our survey results, the average taken across all the years, and looked at what proportion of fields were positive for Aphanomyces plus Fusarium species, and that's shown here in these dark gray to black bars, and then what proportion of fields were positive for Fusarium only, indicating there was no Aphanomyces in those fields, and then the very small percentage of fields that had only Aphanomyces without Fusarium occurring at the same time. And what we can see is that Aphanomyces almost always occurs with Fusarium, but Fusarium doesn't always occur with Aphanomyces. So we're really dealing with 2 root rot issues, one being Aphanomyces root rot with Fusarium, and then the other being Fusarium root rot on its own. However, what we found is that Aphanomyces root rot is really the pathogen that causes the most damage. It's where we see the most yield loss, where we see the symptoms contribute to yield loss. So that's why I'm going to focus most of my presentation on Aphanomyces, but just always keeping your back in your mind that Fusarium can be a problem on its own. And so just a quick summary of knowing these pathogens and what the difference is between these pathogens. When we look at Aphanomyces, some of its key characteristics is that it's a highly specialized pathogen of legumes. And if we compare that to Fusarium, and one of the species that we find most frequently, Fusarium avanaceum, this is a pathogen that is a generalist pathogen and has a very broad host range. So it doesn't just infect, can't just infect legumes, it can infect a number of other crops. Aphanomyces produces these long-lived resting spores that are called oospores. It's how it survives in the soil, whereas Fusarium doesn't produce a long-lived resting spore, and it can survive on stubble and bridging from crop to crop because it has that broad host range. However, it seems that peas and lentils are its preferred host, so it causes the most damage on those, and you might just get very slight infection in some of your other rotational crops. Aphanomyces is favored by warm soil temperatures and excessive soil moisture. And temperatures generally around the 15 to 20 degrees, soil temperatures around 15 to 20 degrees, which is why we see it start to infect around June. Whereas Fusarium avanaceum does prefer higher soil temperatures and drier soils, which is why we see it start to infect usually into July. In terms of symptoms, in a perfect world, we can tell Aphanomyces and Fusarium apart, and I've highlighted some of those symptoms here. Fusarium starts with the blackened taproot, that the infection starts at the point of seed attachment and spreads from there. For the most part, the lateral roots stay healthy, and that means that the roots can still do most of their job. They can get out there, spread out into the soil, and uptake nutrients and water that's needed for growth. And for the most part, the shoots remain healthy. So it's not always obvious that you might have a root rot problem when you're dealing with Fusarium, because the shoots will look really green. And towards the very end of my presentation, I'll show you some pictures from a field trial where we saw that. For Aphanomyces, we see this pinching of the epicotyl that stops abruptly at the soil line. That's a very good indicator that it's Aphanomyces. We often get loss of these lateral roots. They'll show a watery soft brown decay, and eventually they just kind of crumble away to nothing. And we can see that we get a lot more symptom development in the shoot, so it's a lot more obvious that we have a root rot problem. I'm going to go over quickly the disease cycle of Aphanomyces, and then talk about some of the key aspects, and then I'll pause for questions after that. So our initial inoculum of Aphanomyces are these oospores that you can see here. They're very thick-walled resting spore, and if you can make out kind of this double cell wall here, this is why they're so good at surviving in the soil. They're designed to be able to resist most of the harsh environmental conditions like freezing, drying, oversaturated soils. And they will sit in the soil and wait until you plant susceptible hosts, which in this case can be pea or lentils. And in the presence of pea roots or lentil roots, that they will release exudates that induce these oospores to start to germinate. And the oospores can germinate without the presence of water, but they will basically just sit there, partially germinated, until it starts to rain or until there's enough moisture in the soil. And they'll germinate to produce zoospores. And what happens here, as you can see in this picture, this is how they germinate. They produce this hyphae. And then cells start to accumulate here at the tips of these hyphae. And here's actually a picture of the zoospores that are being squeezed up through the hyphae and then accumulate here at the top of this spore structure. They'll kind of accumulate here in mass, and then we call it hatching. I don't know what the actual term is. And then they start to hatch. Well, they start to release the zoospores. And here you can see from a different angle, it's one of these masses of zoospores that started to release, and you can see the zoospores that have swam away from the cluster here. And so these zoospores can swim short distances in water, and that's why they need that saturated soil. And they will swim to find a pea root. They'll infect that pea root all along the lateral roots and the root cortex, and then spread very quickly through the whole root. So you can get kind of oospores to these zoospores being produced to infection within 7 to 10 days after a heavy rainfall. Once they've grown and the pathogen's grown and used up all the nutrients in those roots, then they switch back to this oospore life cycle where they will sit and wait for a susceptible host to be produced again. So from the disease cycle, what we can see are some of the key aspects that start to give us some clues on maybe what management options might be, or how we can start to manipulate the environment so that it's not in the favor of the pathogen. And so some of those key aspects are that oospores require the presence of a host to break dormancy. they will sit in that soil until there's a favorable crop that's planted. The oospores can infect roots directly, and they will certainly do this when there's dry conditions and not enough soil, or sorry, not enough moisture in the soil. But the amplitude of infection will be much lower, and that's because there's the zoospores are the multiplier stage. So you could have one oospore produce about up to 1,000 zoospores, and it's those zoospores that then really drive that high infection rate. The zoospores do require saturated soil to swim to find water. And that's why we see when I showed the survey results, we can see that there was a big difference between a wet year and a dry year. And so what we can see is that the soil moisture and the crop history are really the most important drivers of disease. So I'll pause here for the first round of questions if there are any out there.
So if you check your chat box, you can either enter the questions via the question box, or you can text them to the number that is in the chat. One question I do have that has been sent in is, can high Fusarium in cereals increase the risk of root rot, particularly Aphanomyces in pulses the following year? And was the survey results looked at from a correlation between areas that might have had high Fusarium the year of or the year previous and correlation to root rot?
Yeah, that's a good question. So the Fusarium that causes Fusarium head blight is predominantly Fusarium graminearum. And Fusarium avanaceum can be involved in Fusarium head blight, but it's not the predominant pathogen. So that's fairly good news for this carryover of Fusarium. We do find Fusarium graminearum on pea roots, but at very, very low levels. So it's certainly not the most important Fusarium species. The other question about correlations, when we did the surveys, we tried to collect information on the previous crop, with the most of the previous crop being a cereal or a canola, obviously, and we didn't find any correlations between previous crop and disease level or disease presence. So I think that would indicate that we might have these low levels of Fusarium avanaceum that's surviving on cereal or canola, but it's not, I don't know how to say this, it's not the major contributor. It's still the peas being there that is what causes the pathogens to blossom and multiply on the pea species.
Okay. I think we can continue moving on, and any more questions we can just move to the end as well.
Okay. Okay, so now I'm going to talk about some of the research projects that we've been doing in the lab. And I want to frame these in the context of recommendations that we provide to growers so that it makes sense while we're doing this research. And so our #1 recommendation to growers is to avoid planting peas and lentils in Aphanomyces infested fields, and to get your soil tested if you suspect that you have Aphanomyces, so you can confirm the presence of Aphanomyces. And that's generally been the #1 recommendation for this disease since Aphanomyces has been a problem in other areas around the world. But there's a lot, I think, that can be expanded on that recommendation, and a lot of knowledge that we can gain to try and improve that recommendation and not make it so vague. So we have a little more knowledge that we're working with. And so what we've been trying to do is ask the question, can we quantify the risk levels to come up with a knowledge-based decision support system? And so in order to quantify those risk levels, there's a couple things that we need to know. First is how much inoculum is required to cause disease. The second is, can we actually measure inoculum levels in field soil? Currently, any decision support system or risk index is, wherever it's done worldwide, it's done using greenhouse soil baiting assays, which can be quite laborious and are not that precise. And then the third question is, well, if we move towards a soil testing system and develop a decision support system, where do we need to sample in a field, and how many samples do we need to collect? So that's the basis of the research that I'm going to talk about next. And so, like I said, our first question is how many oospores to cause disease. And this is one of my favorite pictures because I think it really tells a story of what's happening with Aphanomyces and why it wasn't a problem in anyone's radar about 5 or 6 years ago, and then all of a sudden we're finding fields. We're finding that 60% of the fields are positive for Aphanomyces. And so what we can see here is a gradation of inoculum that's been applied to soils in known amounts. And we start here with 0 oospores per gram of soil, move up to 1 oospore, 10 oospores, and then here we have 100 oospores, and then it continues to go up from there. And so what we can see right away is that there's a switch. There's a huge jump from peas being healthy to peas being completely diseased. So this would indicate what we saw in the field, and we're surprised to see so much Aphanomyces because pea fields were doing fine. And then we got into this wet cycle starting at about 2012, 2013, and all of a sudden it seemed like pea fields really were not doing that great. And that's because we needed to hit this threshold amount of inoculum in the soil to see the disease being expressed. So pictures are really nice, and we can see from here that, yes, there's definitely a threshold. And then the question is, okay, what is exactly that threshold? So we go back to good old mathematics, and I don't know if I have ever had to work with a sigmoid curve since I maybe took calculus way back in high school. So it took me a little bit to remind myself of how we deal with sigmoid curves and what some of those equations are. But really, we can see that that's how the inoculum is following this kind of classical curve here, where we have our oospores down here ranging from 0, this is 10, 100, all the way up to 10,000. And the graph shows what we saw in the pictures, that we really don't see a lot of disease down here at the lower levels, and then all of a sudden it jumps up when we reach about 100 oospores. So the question is, and the threshold would be, well, where are we starting to see that shift? So what's our inflection point of this graph? So it turns out, as I said, back in calculus 12, we learned what the equation was for determining the inflection point of a sigmoid curve, so we could determine that mathematically. And then the other thing to note here when we did this study is that we looked at the difference between sterilized soil, so that would be this purple line here, versus soil that was directly from the field that would have a fusarium load already. And we can see that there's a big difference between sterile soil, where we have nothing but Aphanomyces, and then we have this the non-sterile soil where there's other microorganisms at play. So what we found by breaking down and doing some of the statistics is that there is a difference in the threshold oospore levels between the different soil zones, so black, brown, and dark brown. And we also see that there's a difference between sterile soil and field soil. And for the black and the brown soils, you can see that the field soil, the threshold dose is a fair bit lower than the sterile soil. But for the dark brown, we didn't see that quite as much. So in the field soils, we would say that our threshold dose ranges from about 50 to 100 oospores per gram of dry soil. And then, as I said, that threshold is lowered in the presence of fusarium. And we are trying to do some more lab trials now to kind of validate these differences in oospore inoculum doses if we're varying our fusarium dose as well, and how does that change your disease risk. And that's, again, to try and come up with a more precise method for quantifying disease risk and for developing a decision support system. So our next question was, is that, okay, we know what our oospore threshold doses are. Can we use a simple DNA test from the soil to be able to measure these threshold doses? So I'm going to show you the results there. And on this graph here, what we have is our actual oospores. So this is what we added to the soil. And then here on the y-axis, this is our measured oospores. So this is what we measured back using our DNA test. And if it was a perfect relationship, we would expect the line to fall somewhere along here. And so I'm going to break this down for the 3 different soil zones. And what we find for black looks pretty good. You can see that the line is following pretty close to our expected line here, with a little bit of maybe some false positives at the lower end here. But in my opinion, it's better to have false positives than false negatives at these lower doses, and we'll get to that later on in the presentation. When we look at the brown, we can start to see that the line is starting to fall further away from our perfect relationship here. We're still, you know, these lower doses we're still picking up, but we're starting to see that our percentage difference from our what our actual is quite a bit lower. And if we look at our threshold dose here of about 100, we're actually measuring a lot lower with that DNA test. And then finally, for the dark brown, we can see that the line is falling much below our expected line here, where we have a lot of trouble detecting at lower levels, and then we're always underestimating what that OOS4 level is. And this is partly because dark brown soils are very high in clay content, and clay can really mess up a DNA extraction. DNA really likes to bind tightly to clay, and so you're removing that DNA out of the test for analysis. And so we're working on now, one of our major activities right now is looking at ways to improve DNA detection, particularly from the dark brown and the brown soils, so we can have a more precise and accurate DNA test to try and quantify risk. And so then the next thing that we looked at was the distribution of Aphanomyces within fields, with the question being, well, how many samples do we need to take to be able to accurately identify fields, and where's the best place to sample from? So what we did over 3 years, tested about, I think it ended up being 55 fields, is that we would just go out into a field, talk with the producer, ask them if they could identify a hot spot where they thought that they had root rot. We'd start there and then start to just sequentially move out for a total of 11 samples within a field. We take a soil punch and then break that up into the 3 different layers and tested it 2 ways. The traditional greenhouse bioassay method, which is really a lot of work, takes about 4 weeks to get results, and then our fast DNA extraction method from soil, which takes less than 24 hours to get results. And we added, besides just looking for Aphanomyces, we added Fusarium Avanasium and Fusarium solani to the DNA test, so we could test for 3 pathogens all at once. And then I broke down the results to look at the field prevalence and the mean site incidence. So what we see from our greenhouse bioassay results, and this is looking at the 3 different depths, 0 to 20, 20 to 40, and 40 to 60, is that we definitely have the highest level of Aphanomyces inoculum in that top 20 centimeters, and that it goes down from there, but there still is Aphanomyces inoculum in that lower level. And then these pictures here just show the difference that we saw in disease severity, very high disease severity at that top level, and then it gets better as you move down the soil, the vertical soil profile. And then in terms of the DNA test results, this is where it's nice because we can actually distinguish what's Aphanomyces versus what's Fusarium, so we get a little more information out of that. We can see a very similar pattern. Here we see Aphanomyces, we had the highest positives in the top 20 centimeters, and it goes down from there. And then interestingly, for the 2 Fusarium species, we can see that it's very high on that top 20 centimeters and extremely low in those bottom layers. So Fusarium seems to be more concentrated at the top layer, doesn't move down to those lower levels, whereas Aphanomyces, the bulk of it is in that top layer, but we still find it in those lower levels. And then the other thing I want to talk about here is the horizontal distribution. This is a little bit harder. It's a little bit harder to wrap my head around and figure out how we can summarize the results from 55 fields and the distribution patterns of Aphanomyces without sitting here and showing individual slides for 55 fields. So what I've done is broken it down to provide a snapshot of 2 fields that showed very different distribution patterns. And then in the end, I will provide a summary of all the 55 fields. And so what we saw in one field, or this was one type of field, was that when we looked at a site from 1 all the way to 11, all of our 11 sites, we saw very high disease severity. And this is showing, the gray bars here are showing the results of our greenhouse bioassay and the disease severity. And our rating scale goes up to 5 here, and we can see that that a lot of those sites are almost at that maximum level of disease. And so we also did the DNA test at these sites, and I'm going to show that here with these black diamonds. And what we can see is some of those challenges with the DNA quantification and detection of Aphanomyces. So all of the sites were positive. So we have You know, we're above 0 for all the sites, so that's good. It means it's matching what we found with our greenhouse bioassay. So we have 100% detection, but we can see that the levels of Aphanomyces here, and on this axis, it's number of oospores per gram of soil. We can see that it really varies. And if we're looking at a black soil here and our threshold is 50, we can see that some of those sites are showing us above that 50 threshold, but a lot of the sites are showing us below that 50 threshold. So again, just an indicator of the challenges of trying to use a DNA test for quantification. We can use it for detection, but we need better sensitivity and precision for quantification. And then just a visual of what this field looked like. So basically we started here sampling at 1, GPS tagged everywhere where we stopped, and we can see that every stop along this field was positive for Aphanomyces. And from the greenhouse bioassay here, we can see those very classical symptoms of Aphanomyces and very high levels of disease. And so in contrast to that, some fields look more like this, where we're looking at our greenhouse disease severity here. And these I would all actually, anything that's under 2 or close to 2, we would classify those as being negative for Aphanomyces. Based on the symptomology, it was pretty clear that this was more of a Fusarium issue than an Aphanomyces issue. And really that there was only one site here that was positive for Aphanomyces. And so in a way, this was encouraging because it means that we can pick up, even in a field that is not heavily infested with Aphanomyces, we can still pick up a positive site, given that we sample enough sites within that field. And then when we look at our DNA test, again, fairly encouraging results, where almost all of them were negative for Aphanomyces, except for that one site that was positive. We did pick it up as being positive as well, but again, at a fairly low quantification level of only about 20 oospores per gram of soil. And then if we look at that, a picture of what that looked like. So we started sampling here, kind of at a high spot in the field, sequentially sampled. This was our one positive, and you can see that it's associated with a fairly low spot in the field here, and then moved on to negative sites. And then here's a picture of what that looked like, that one site that had very high levels of disease, and then the rest of the pea plants that were grown in the soil were very healthy. So I think, again, this was encouraging to say, okay, we can sample, we can pick out a field that has a fairly low infestation level, and we can get a positive and know that there's going to be, that this field needs to be watched as a potential risk for Aphanomyces developing. So when we try to break down the results or summarize the results across all 55 fields, what I've done is broken it down for the percent of fields are shown here on the y-axis, and then breaking it down by the percent incidence or percent positive samples within each, for each field. So the categories here were 100%, so that would mean all samples tested from that field were positive, and then we have 75 to 99%, 50 to 74, and so on. And then I'm going to show you the results of the PCR compared to the greenhouse test. And so what we find first for our first category here is that about 70% of fields, when tested by the greenhouse method, had 100% of their sites positive. Compare that to a fairly low number with PCR, about 20% of those fields showed that all those sites were positive. When we look at 75, we're picking up, we have more fields that end up falling into this category where we had less with the greenhouse, and it continues in that same pattern. So what we can see is that the spread of positive samples when we test by PCR is a lot larger than the spread for greenhouse. And so again, this means that we can detect positive samples very nicely with PCR, but we're maybe not detecting the right amount of positive samples. So our incidence levels are off, but if we look at prevalence, both with PCR and greenhouse testing, we would say that 100% of the fields were positive. And really wherein the problem lies, and as I showed you earlier, is that it is the ability to detect these low-level fields that maybe don't have as many positive sites, that is a little more imprecise with a DNA test than a greenhouse test. So from this project, we were able to come up with some recommendations for soil sampling. And these are to remove the stubble layer and then take soil from the top 20 centimeters from low spots in the field or with history of yellowing the last time you grew pea or lentils. So it means that in order to sample, you have to be aware of your field history and remember where you maybe saw that peas or your lentils didn't yield as well. But that if you target those low spots or areas that you know have been problematic, you are likely to get a positive result. We have also found, and I didn't get into these results, that pea or lentil post-harvest soil sampling does tend to give better results than a preceding spring sampling. So that means that after you've grown your pea or lentil crop and you notice that maybe you're starting to develop a few yellow spots, that's the best time to go in and sample rather than waiting 3 or 4 years until you go into that field again and then deciding to sample. And I think this just has to do with the oospores. As they go into dormancy, they become more and more difficult to extract the DNA from. So fresh oospores tend to give us better results than aged zoospores. We're still working out this sort of minimum probability at the minimum number of samples that are needed in order to make sure that you get the correct result. But I think as I showed with that field where we sampled 10 sites, we were able to pick out one positive site as long as you're targeting those low spots. And then just a word of caution here, do beware of false negatives and ask questions about the analysis method. And most of the soil testing labs are testing using DNA, and that's fine. That is the most efficient way to do it, and it's the way you can get the quickest results. But if it comes back negative and you were pretty sure that you had a problem in your field, maybe ask some questions about the analysis methods and make sure that there was the correct standards in there to validate your results. So just for the summary of the research portion here, we do find that the oospore threshold to cause disease is low. It's lower than for other soil-borne pathogens. That's because of the nature of the disease cycle where the zoospores multiply from the oospores. And because that oospore threshold is fairly low to cause disease, it does make the DNA testing a bit more challenging because we are looking for fairly low numbers in the field. And so the DNA test is very good at identifying high-risk fields or those fields that have a very high oospore load, but it is still difficult to detect at those low oospore levels. And that's because we're basically looking for 50 cells or less in a gram of soil, which really isn't a high amount. We are working on methods to try and improve oospore detection that maybe involve starting with a larger amount of soil, trying to remove or filter out those oospores, and then apply a DNA extraction step to the oospores rather than the soil. And then Aphanomyces eutyches is highest in the top soil layer, so we do recommend sampling from that top 0, 15 to 20 centimeter layer, but we do find it in all layers, which again is an indicator that it has likely been present in our soils for a number of years for it to be able to trickle down that far into the soil profile, but it's the stuff that's in that top layer that's causing the disease problem. So with that, I will pause for questions.
Thank you, Syama. You've been doing a lot of work with the soils, and we've learned a lot in the last, 5 years, I guess, since you've been doing this. So thanks so much for all your work. Some questions that have come in. How and when do you get your soil tested? How and where, sorry, not when. How and where do you get soil tested?
So most of the the seed testing labs do offer an Aphanomyces test. I know in Saskatchewan, if I can remember all the labs offhand, like Discovery Seed Labs, I think provides tests. So does Quantum Genetics. Sherilyn, you probably know what all the labs are that offer it, but yeah, most of them offer it. Some of them offer a DNA-based test. Some of them are offering a soil grow-out test, and they all have instructions on their websites on how to collect the soil and how to send them the soil. And I think it typically involves sampling at a few different sites, again, targeting those low spots. And then you can bulk the soil from a few sites within your field and then send it to those labs.
To add to that, there is an article that that is on our website, on Saskatchewan Pulse Growers website, on testing soils for Aphanomyces, and it lists all the labs. It's also in the root rot document that is available on the provincial Pulse Growers websites as well. Adding to that question, is there a difference in the soil tests that one can get done? And what do the results really tell us?
Yeah, so most of the labs are offering the same DNA-based test. So it means they're using the same barcode to be able to identify Aphanomyces. And there's a couple of labs that are doing soil isolation or soil greenhouse grow out. And I think if you look around on their websites or look at that list that Sherilyn said, that might be clear which labs are doing what. And I think what the results tell you is if you are positive for Aphanomyces, then you should avoid planting peas or lentils in that field for a number of years. And what we're trying to do, and I guess that's this whole idea behind a quantification test, is to provide more in-depth information or results on that soil test, because right now all it's telling you is presence or absence of Aphanomyces. We're hoping to be able to, if we can quantify it and you know that you're just below threshold levels, then it means that you could maybe apply some of our management options that I'm going to talk about in the next section, where management options seem to have better promise in these low inoculum fields than they do in very high risk fields. So that is one of the aims of a soil test. And I guess the other one is so that you know when it's safe to plant peas or lentils again. If you know that you have Aphanomyces or you've tested your soil and it's positive for Aphanomyces, you waited 4 years and you want to know, well, can I plant peas again in this field, or am I taking a risk? If you can get your soil sampled again then and say, okay, the risk has gone below threshold, then you know that you can plant peas or lentils again. So I think with our research, that's what I'm aiming towards. We're not quite there yet. It's going to take a couple more years of really trying to fine-tune this oospore, the DNA extraction from oospores. But just for now, in terms of the presence or absence of Aphanomyces in your soil, I think it helps make those decisions on rotation and what pulse crops you can grow in your field. Thank you.
There's another question related to soil sampling. And the question is Aphanomyces photostable? There's been some discussion on making sure that the soil is taken from the 3-inch to 9-inch mark and avoid soil surface. Is that now incorrect with further research? And I guess as it relates to the photostability of the Aphanomyces.
Right, yeah. That's a good question. I think for the most part, that top layer of soil is covered by stubble anyway, particularly if in a no-till field. So you wouldn't get a lot of UV or UV kind of penetrating through that stubble layer anyway. Yeah, and with the data that we looked at, and we didn't worry about removing it the top 3 inches, and we had no problem detecting it. So I think you're safe to just go from the top, the zero, like right below the stubble layer, down to about 15, 20 centimeters.
The last one regarding kind of the soil testing, and then we'll let you get moving on, is do you see the same separation between the autoclave soil and field soil when you treat the seed with a fungicide that controls Fusarium? For example, can you control some of the Aphanomyces by treating the Fusarium?
Yes. So when we did that test, the greenhouse test, we did actually treat all the seeds with a fungicide against Fusarium, because we just wanted to be able to identify Aphanomyces symptoms. But when we did the DNA test, that was on fresh soil. I know, a little bit confusing. Then there was no effective fungicides on there. So we would see that those fungicides on the Fusarium, the fungicides against Fusarium are definitely work to exclude Fusarium from the system. I don't know if that totally answers the question, but.
I think it helps.
Yeah.
Because it was relating to the, autoclaving the soil versus field soil, and just there's an impact if you used a treatment, which you said you did, right?
Yes, so. Yeah, we did, yeah. And some other research that I didn't get into here is that we have been looking at that interaction between Fusarium and Aphanomyces, and the root rot severity and the speed with which the peas die is much faster when both Aphanomyces and Fusarium are involved. And so adding that seed treatment against Fusarium does help to mitigate that synergistic effect.
Okay. Thank you. We'll let you carry on now.
Okay. So the last section I'm going to talk about here is management options, and again, kind of framing those with some of the field trials that we have been doing over the past number of years. And so again, continuing with this idea of what are our current recommendations to growers. So in addition to avoiding infested fields, the other thing that we suggest is a prolonged rotation away from susceptible hosts. And this includes peas and lentils in infested fields. And then also consider using a seed treatment that targets the root rot complex. And we just discussed that a little bit with relation to Fusarium. And so some of the questions that come out of these recommendations then is, well, how long is long enough? I think we often say 6 to 8 years, but that is maybe a bit of a guess based on what they found in other areas, not necessarily fitting in with our prairie conditions. And then the other question is, well, if you're taking peas and lentils out of a rotation, can we add other pulse crops there that maybe can be grown instead that won't contribute to the problem? So we did some field trials there. And then the other thing that with this idea of seed treatment is, well, which ones can be used and are they actually effective? So that's what I'm going to focus on today with some of our results. And then I think on my last slide, I have a list of whole bunch of other field trials that we're also doing that I just didn't have time to talk about today. So in terms of field trials, I have a picture here of what one of our trials looks like. And one of our big challenges with field trials and working with Aphanomyces is that we cannot inoculate it in the field and get disease to develop. I think it has to do with the time that it takes to reach that threshold dose. So we have to go out and do all of our field trials in producers' fields that we have identified as having Aphanomyces problems. And we picked out a lot of those from doing our surveys. So everything has to be done off-site. And so you can see here, this is one of our field trials, and we're actually in the middle of a producer's field that was growing mustard. So we kind of have our little island of peas, and then we're surrounded by whatever crop the producer's growing. And so, like I said, the challenges are that these inoculated field trials do not work. It's frustrating that a pathogen like Aphanomyces can cause so much disease, and then we're trying to get disease, and we tried everything we can to get it out in the field, and we just can't get the inoculum to take. And so, like I said, we do them in producers' fields, but the problem with this is that there's no healthy check. Most of these field trials related to disease, you always want to be able to use your not diseased or healthy check as a comparison, particularly to start looking at what some of those yield impacts are. And because we can't have that healthy check, it's hard to define exactly what that yield reduction is. But we always kind of look at a silver lining, and what we found is what's been helpful is that it is providing an opportunity to track changes over time out of peas. And so that we can go back in the producer's field and kind of move our trials around and then keep track of their rotation. So we're kind of building a little bit of a rotation component into these field trials as we move them around in a producer's field. So what I'm going to start out with is showing some of that data from 4 years of field trials and how we found that Aphanomyces levels change or don't change over time. And again, we have about 6 or 7 sites across the province, primarily in Alberta, and we have a couple sites in Saskatchewan. And I'm just going to focus on 2 sites so that I'm not here for the rest of the day. So I'm going to start with Drumheller, which is really one of our worst sites in Alberta. And they last grew peas in 2014 at this site, and were in the situation where they basically did not get a yield off of the field as well. And so we went in 2015, started our field trials, and have been doing them there for the last 4 years. And the other thing that I've done just to try and kind of match it with weather patterns is shown what the rainfall, total rainfall was during the growing season, and just compared that, what the difference was from the long-term average for that site. So what we can see first is that in 2015, when we tried to go back-to-back straight into peas, we didn't get any peas growing at all. So we weren't able to test disease severity and we got no yield. The 2016 up to 2018, we've at least had peas established in the site, but we can see that disease severity has remained fairly high. We saw a bit of a dip in 2017 when it was really dry, but then if you remember that the 2018 season started out very wet, heavy snowpack, lots of spring flooding. So we started out the season fairly wet and then it dried up as the season went on. So that's why we still see fairly high disease severity. And then in terms of yield, I guess it's a little bit encouraging, although the yields aren't great. But I guess what we can see is that, you know, we really didn't get any yield those first 2 years, and that yield is starting to get better, even though the root rot levels haven't really got better. So we seem to see some recovery of yield before we see recovery of disease severity. And just some pictures of what that site in Drumheller looked like. This was in 2016, which was a very wet year. We were also on a slope here, so we can see down here at the bottom where the water tends to accumulate, very, very poor pea establishment, and it does actually get better as you move up slope. And the other thing that we started doing in 2018, and this is looking at the interactions between Aphanomyces and Fusarium, is we did start to track how the pathogen composition is changing on the roots over time. So we went in and collected samples in June, kind of mid-June, and then we went and collected samples again in early July during flowering period. And what we can see here, we have Aphanomyces and then 3 different Fusarium species. You can see that when we test in June, we have very high Aphanomyces levels on those roots, and then that really drops off by July, and that's because that's when Fusarium starts to move in. And remember, right at the beginning of the presentation, I had said one of the big differences between these species was their favored soil temperatures. And so we see Aphanomyces come in when the temperatures are a bit cooler, and then Fusarium come in as the temperatures are kind of coming after Aphanomyces when the temperatures warm up. And just showing pictures of that. These are samples collected in early June. You see mostly the Aphanomyces symptoms, and then collect plants again 2 to 3 weeks later, and we can see that the blackening of the Fusarium has moved in. And so part of the problem is that historically root rot surveys are always done when peas are flowering in July. And so we would have pulled out peas, looked at something like this, and thought immediately that it was a Fusarium root rot and not an Aphanomyces problem. So that's another reason why we're kind of Aphanomyces seem to explode in 2014, and it's because we are probably missing a lot of it because of the timing of sampling. And then another site, this one's also in Alberta, Penhold. This is near Lacombe, and this is our black site. This one is nice because I actually last grew peas in 2011. Again, had a very poor pea crop. I think the producer said he wasn't going to grow peas again there. And so we came in a little bit later already coming into this field. And so what we see for disease severity is that even 4 years later, we saw a pretty high disease severity. On this scale here, our max is 7, so we're just about at the max. But we can see that that disease severity really started to decrease over time. And then this was an interesting site because even though we have these really high disease severity, we still managed to get fairly high or very high yields off of this site. I know small plots tend to give us higher yields than they do out in commercial sized fields, but still I was very surprised to see these high yields when we had root rots at these levels. And then we see that the yields really improved, and we're seeing probably maximum yield that you can get as that root rot level starts to decrease. So I think this is an encouraging result showing that 2011 to 2018, so kind of that 6 to 8 year period that we have been recommending seems to be on target, at least for some fields, and this field particularly in the black soil zone. And I just want to show you a picture again of what that looks like. And this is the plots in 2017. Beautiful green, lush plots. You can see the amount of pods on these plots. We thought that there was no root rot at all. We pulled the, oops, a little too far. pulled those roots out, and it was surprising to see that this is what the roots could look like this, and we can still get these phenomenal yields. So there's an interesting dynamic between kind of levels of root rot and yield potential that has been really hard to try to figure out, and that's partly because we can't do sites, do trials in sites with a healthy check. Okay, so I'm going to move on and talk about two other projects that we're working on. This one has just started. This one is looking at the effect of rotation with a non-host pulse crop. I know lots of stuff on this slide, but this is basically our treatment layout for the next 5 years. And what we're looking at is different numbers of breaks between a pea crop, and then if we replace that pea crop instead with a non-host pulse crop. And so we started out with just a pea and then a cereal, and then this year in 2018, we're looking at effects of different pulse crops. We're looking at chickpea, faba bean, and soybean as our 3 options for non-host pulse crops, and then at 2 different locations, one in Saskatchewan and one in Alberta for each one. And then we added a location in Manitoba for soybean because there's so many soybeans grown in Manitoba. And so we'll kind of continue with the peas, extending that pea rotation, and then adding in different non-host pulse crops at different points in the rotation. And so I'm going to show you the results from 2018. And first, what I want to highlight over here is the results of our Aphanomyces numbers. So this is number of cells or oospores per gram of root when we look at the pea crops at our different sites. And we can see Lacombe, this is that site that I just showed you that really had very little disease. So we're getting very few oospores being produced on those roots. And then it kind of ranges. This is Saskatoon, fairly low. Swift Current is fairly low. And then we get to these extremely high numbers here in Lomond and in Redvers. And this one I actually didn't even take the bar all the way up, but 325 oospores per gram of roots. And I think that's just an indicator of showing how these oospores can get left behind in the soil. And then we compare it to all of our non-host crops here, and we see very, very low numbers of oospores being produced on the roots, and most of these are not significantly different from 0 when you do the stat. The only one that we kind of saw here was chickpea and Swift Current. We did see it support a few oospores on the roots, but nowhere to the same extent as pea. And I think chickpea, there might be differences in varieties. This one was Orion, this one was Leader. So we are going to do a little bit of further investigating to see if there's differences in varieties on chickpeas and ones that are maybe can still host, can still maintain a little bit of Aphanomyces versus ones that don't allow any growth at all. So that was just the first year of this trial, and we'll continue with that. And then the last thing I want to talk about is some of the field trials that we've been doing to evaluate different chemical products. And we've done these for a number of years. They've kind of changed every year, kind of depending on what was available, but the one that's stayed consistent over the past 2 or 3 years is looking at the effect of edge at the recommended and 2 times the rate, different seed treatments at both the recommended and 2 times the label rate, and then with and without phosphoral. And the idea was to look at whether we could build a package that built on all 3 of these products. And I'd say the results were not great, and I'm going to show you results just from some sites where we saw some significant effects. And interestingly, we haven't seen any significant effects of any combined treatments. It's only been kind of single treatments where we see an effect. And so I'm going to show you results from Scott, Saskatchewan from 2018. And I think this was the 3rd year we did this site in did this trial, and 2018 was actually the 1st year where we saw an effect of a treatment. And what we saw was that it was an effect of the edge. We did see higher yield compared to the label rate, but no difference in disease severity. So this is likely more of a weed suppressive effect than a disease severity effect. For phosphoral, which is applied 2 times in the growing season, again, we saw a significant increase in the yield here, but not really a big difference in disease severity. So that could indicate that that effect was more to do with plant health effects. And then finally, with the seed treatments here, this one is a non-registered product, so I'm not allowed to say what it is yet. And then we compared it to ethoboxam at 2 different rates, and then just our check with no seed treatment. And we can see that we did kind of get a little bit of a yield boost at 2 times the label rate of ethoboxam. We did get a reduction in disease, a significant reduction in disease severity compared to the check. And then this other product actually performed fairly well, and we did see a yield increase at that at 2 times the label rate. It'll be up to the company whether they want to do further testing or pursue registration on that product. And then just another location, Tabor, where we saw much higher disease levels than in Scott. Again, we see an effect of that 2 times edge, and here we actually did see some significant differences between disease severity. But you'll notice that our yields are much lower in Tabor compared to Scott, like I said, because the disease severity numbers are quite a bit higher. And then in terms of the seed treatment, again, we saw this product give us significantly higher or significantly better disease or significantly reduced disease severity compared to these treatments, but we really didn't see any difference in yield. And again, we have this, this trial replicated in 6 or 7 sites, and we saw kind of different results at each site. And so what, I guess the take-home message here is that the seed treatments can be effective at sites with a lower disease pressure, which again would feed into this idea of developing a decision support system where we could quantify and know what our risk of disease is. So in summary, for field management, this will wrap it up. Do test your field if you suspect Aphanomyces root rot, but make sure that you're asking questions about that testing method, and just be aware that false negatives can occur. Seed treatments do provide some protection, but not in the high-risk field. And what we saw that was interesting was that the root rot decreases and yield increases with length of time out of P, but it seems like there's a yield response quicker than there is a root rot response. So we start to see that the root rot severity levels don't change, but we still get a better yield, and I'm not quite sure why that is happening yet. And then lastly, there is lots of research that I didn't talk about today. I think I'm already a little bit over my time. And here's a list of other things that either I'm working on or working on with collaborators. That includes breeding for resistance, looking at cover crops, looking at intercrop. We're trying to look at different environmental conditions that affect the survival of oospores, and maybe the ability to detect them will depend on environmental conditions. I mentioned briefly we're doing a lot of research looking at the interaction of Aphanomyces with Fusarium and how Fusarium is contributing to the root rot complex. We're going to be starting to test some biocontrol products, and I know that there's other companies out there that are testing biocontrol products. And really my ultimate goal is to be able to develop this decision support system that is based on quantification of oospores in the soil and kind of knowing your risk. So I'll just end there with thanking the different collaborators that work with us on field trials, all the technical assistance that we get from technicians and co-op students, and then finally the funders over the past 5 years that have really been able to push this research forward and I think get a lot done in a short amount of time. Even though research takes a lot of time, I think definitely the amount of knowledge that we've gained in the past 5 years, we're at a much better point now. So with that, I will leave it for questions.
Great, lots of information and lots more to come yet. In terms of questions, there's a few that have come in. The first one is, what are the other crops to be watching for in the rotation that are host to Aphanomyces? Peas, lentils, alfalfa, witch clovers, any other crops or weeds, I will add, to be considered?
Yes, yeah. So there's a lot. So I guess I went over the ones that you don't have to worry about, and that's pretty much chickpeas, faba beans, and soybeans. All the other legume crops can likely be a host. Alfalfa can be a host, but it does depend on the variety. There is a race structure to Aphanomyces, and we don't know which race we have. So there's race 1 and 2, but it relates to alfalfa, not to peas. So all races can infect peas, but some races can infect alfalfa, if that it's complicated, if that makes sense. But I don't think we know here in Canada, our alfalfa varieties aren't tested for Aphanomyces, so we don't necessarily know which ones are resistant or susceptible. I think there's a researcher out in Quebec that is doing some research on that. The other legume forages can be a host, particularly the vetches, which I know are becoming more popular to use as a cover crop species. Clover also have variable response. Some clovers are susceptible, some are not. We are planning to source a lot of these forage legumes this summer and just grow them out at one of our highly infested Aphanomyces sites, so we can get a little more precise information on which ones are hosts and which ones aren't.
Thank you. Another question revolves around one of the slides that you showed earlier versus later sampling time of peas, where there's Fusarium versus kind of the Aphanomyces. There were notches on the leaves suggesting pea leaf weevil. So the question is, how does pea leaf weevil impact the potential disease or root rot disease severity, and what should we be doing to?
Yeah, that's a really good question. We did some research on that. We did both lab, greenhouse trials and field trials. In the greenhouse, I feel like I deliver nothing but bad news sometimes. But in the greenhouse, we did find that pea leaf weevil larval feeding is synergistic to root rot, and vice versa. So the pea leaf weevil seemed to do better when root rot was present, and root rot also seemed to do better when pea leaf larva was present. So there's we hypothesize there's some sort of compensatory response happening in the peas, so it actually makes the nodules a little juicier for the pea leaf weevil larva, and then a little better for root rot to infect. So I said, I always feel like I'm the bearer of bad news. But then out in the field, we tried to do kind of replicate what we saw in the greenhouse and do field trials. And these were based on using seed treatments to try and exclude, Fusarium, or try and exclude the pea leaf weevil. And the seed treatments are really good at excluding the pea leaf weevil, but they're not as great as excluding the pathogens. So what we saw was that, again, a complicated story. I would say that out in the field, we didn't see that same synergistic relationship that we saw in the greenhouse, or not to the same extent. In the greenhouse, it was extremely obvious, like the data was very, very clear, whereas out in the field, it was a little more muddied as to whether we were seeing that same synergistic effect. But I think in the field, when you're using a seed treatment against pea leaf weevil, it works fairly well, and so that at least will help to reduce that effect that can be occurring, or the interaction that can be occurring between the two.
Okay, thank you. Another question is, how is the seed treatment like Intego Solo from New Farm reducing the severity of Aphanomyces?
Yeah, so those are the results. I should have said the trade name. So that's at the box is Intego Solo. And what we see is that we do see it have a positive effect at our lower inoculum sites. So the sites where we've been out of peas for longer, or where we just started with a lower inoculum level, we do see that there is an impact. Now, Intego Solo is registered for early season suppression of Aphanomyces, and I think the results and the data that we collected supports that claim, right? We see an early season suppression, but when we start seeing the root rots come in later in June, if that's when we get our heavy rains, then that seed treatment doesn't really have any effect anymore.
Okay. And I think the last question we have coming in is related to Alberta. What are the other root rot pathogens that could affect pulse production in Alberta? But I think we can relate that across the provinces.
Yeah. So in terms of peas and lentils, Fusarium and Aphanomyces are definitely our number one targets. And then if we start looking at the other pulse crops, I think another unfortunate part of the story is that fava beans, chickpeas, and soybeans are all fairly susceptible to Fusarium. So you're not going to have the Aphanomyces issue, but like I said, Fusarium root rot is a disease on its own, and so you might still have a Fusarium issue. And I know that it can pop up in faba beans and chickpeas from time to time. I think the good news with chickpeas is that a seed treatment does a fairly good job of reducing Fusarium impacts at seedling, and that's when chickpeas are the most susceptible. But you still can get Fusarium root rot later in the season. And then we have all our other regular players like Pythium and Rhizoctonia. Pulse crops are all very susceptible to those pathogens. Those are also usually taken care of with a seed treatment because they're more seedling blight issues than late season root rot.
Okay. One final, sorry, I have to apologize. One final question.
Okay.
And this is one that I often get is there a difference between the susceptibility of varieties that have, say, a tannin in their seed coat to the root rot organisms? Things like the maple peas or the dun peas versus like a yellow pea? Yes,
yeah. There is the dun colored or the maple peas, the ones that have the tannin seed coat, do tend to be less susceptible to the root rot. It appears, we've done just a little bit of work looking at timing of susceptibility. It appears that it helps them more at the early seedling stage. And then once those tannins have kind of dissipated from the seedling and you're just down to regular roots, they are equally susceptible. But having that little boost at the beginning seems to help them carry through the season. So there is definitely truth to that observation that the dun-colored maple peas are less susceptible to root rot.
Okay. Thank you. I think that about wraps up the questions. So thank you very much, Dr. Chatterton, for everything that you've presented here. And I can see in the future we're going to need root rot part 2 as the next, you know, all the data that you're going to be developing and have available in over the next few years will be great. Yeah. So just a reminder for those that have participated that an email will come to you and you can reply with your CCA number if you are a certified crop advisor, and that will give you your credits. You can also provide suggestions for future webinar topics at the same time. Feedback and suggestions are always appreciated. Thank you, a big thank you to Dr. Chatterton for speaking today to us on root rots and pulses, and Andrew for organizing the event, and a big thank you to all the participants for joining us. Our next webinar we have scheduled for June 5th will be Dr. Barb Zeisman, who is the Provincial Plant Disease Specialist here in Saskatchewan, and she is going to cover integrated pest management for diseases and pulse crops. So for more information and registration, you can check out our SAS Pulse website under the events section. So with that, we can end the webinar, and thanks again to everyone for joining, and please have a great afternoon.